Animal viruses. Animal viruses means a virus that is going to infect the animals. It is going to consist of protein shell, which is known as capsid, and DNA as a genetic material. It can be a DNA or RNA, but most of them are DNA that is packed inside the capsid. Next, we are going to see about one of the examples in the animal virus, that is influenza virus structure and the replication to be taken place. They are negative sense strand, which means a positive strand DNA is going to synthesis another DNA for replication. After that, it undergoes transcription to RNA. Then the RNA is going to synthesis protein by translation. If the strand is positively. But in the case of negative sense single strand, then the negative strand DNA, RNA or a DNA is going to undergoes synthesis of complementary strand for the negative strand. After the complementary strand is synthesized, then the translation of a protein is going to be taken place. That's the differences between negative sense and positive sense. An influenza virus genome is going to consist of 13,588 base pairs long. It's going to code for 10 to 14 proteins. The each proteins we are going to see its encoding protein and its function. In general, this is the influenza virus structure. It's going to consist of 8 segments of genetic material. It is going to be a 1, 2, 3, 4, 5, 6, 7, 8. So it is the 8 segment of genetic material in the influenza virus. So each of the segment, what is its function or all we are going to see. So first segment is going to be encoding the PB2. It is known as PB2 gene. It is going to be a first gene is going to encode the PB2 protein. This protein is very important for polymerization, which means adding of nucleotides to the 3' end during the replication is going to be carried out by polymerase enzyme. This polymerase enzyme subunit is going to be coded by this protein PB2, going to be done by this protein PB2, which is coded by gene 1. It is also involved in mRNA cap recognition, where PB1 is going to be synthesized by second segment of gene. This protein is going to involve in polymerasive subunit of RNA elongation, which means it is also a polymerization process where adding of nucleotides at the 3' end. It also involved in endonuclease activity, which means creating a nick by cutting out the nucleotide bonds. Phosphodiester bonds are going to be cut off by endonuclease to create a nick. Next, a third segment is going to encode the PP1F2, which is involved in pro-apoptotic activity. Due to mutation or any of the processes which causes the apoptosis or lysis of the moles is going to be carried out by this PP1F2 protein. Next, a PA protein is also synthesized by the third segment of a gene. It is going to involve in polymerization action and protease activity during the lytic cycle. Next, a fourth segment is going to be synthesis the protein HA, which involve in surface glycoprotein synthesis. Surface glycoprotein is a capsid protein which is found above the genetic material, is going to be synthesized by HA protein. NP protein is going to be a nucleoproteins. These proteins involve in binding to the RNA and nuclear import regulation, which involves importing of nucleoproteins, is going to involve in importing of nucleic acids. It's going to bind to the RNA to be synthesized. After it get attached to the RNA, it lead to arrangement of protein over it, capsid protein over it, and causes its packaging. Na is going to be a neuraminidase. It is also a surface glycoprotein, which is very important for viral release, whereas a glycoprotein is very involved in entry of virus. There is a two proteins, capsid protein HA, which is going to synthesize the glycoproteins. These glycoproteins are involved in entry of virus into the human, that is host. Whereas Na is going to be a neuraminidase protein. After the replication to be taken place, it gets released off. The releasing is going to be carried out by neuraminidase protein. Next, a M1 protein, M2 protein, both are matrix proteins. It is going to involve in viral budding. A viral budding after the replication taken place, it's going to released off by budding, because influenza is going to carry out the non-lytic cycle. So it involved in budding. The M2 is going to be present in the matrix, which involve in assembly of genetic material with the capsid proteins. Next, NS1 is going to regulate the host gene by stopping the replication of the host gene and increasing the replication of the viral genome. Where NEP protein is involved in nuclear export of RNA. So here we can find that, as we saw before, influenza is going to be a single stranded RNA virus of negative strand, which means a replica is going to be made. A positive strand is first synthesized, and from this RNA positive strand, a translation is going to be taken place by taking up the machinery of a human. Therefore, it is a genome, 8 genomes. As we saw before, each of the protein synthesized by this genome. So, a influenza polymerase is a enzyme which is going to involve in polymerization action of RNA. After the polymerasive action, a translation to be carried out from the polymerized and newly formed RNA segment. A nucleoprotein is going to carry out the nuclear export. Then the viral RNA. These are the viral RNA. Then a hemagglutinin. Hemagglutinin is going to be a glycoprotein which involved in entry of virus into the host. Where a neuraminidase is going to involve in exit of virus from the host by budding. A membrane protein, where there is two membrane protein, M1 involved in helping out for the budding of virus, and M2 was involved in assembly of protein. A nuclear export protein involved in export of viruses. Next, we are going to see about the plant viruses. Plant viruses are going to be RNA viruses. 75% of plant viruses has 5S RNA as a genome, and 65% is going to have 15S RNA, and 10% are going to have a 5S RNA. Then 5% is going to have a double-stranded RNA. Very few plant viruses are going to have a DNA. So, most of the plant viruses are going to be RNA virus. Now we are going to see about one of the examples in the plant viruses, that is tobacco mosaic virus, which is the RNA virus that is going to be single-stranded, which has positive sense. As we saw before, a negative sense and positive sense differences. As positive sense, RNA can undergo translation directly, where the negative strands have to synthesize its positive sense by polymerization action. Then through its positive sense, it's going to undergo the translation process. Tobacco mosaic virus is going to infect a wide variety of plants like many of the solanum species, and especially a tobacco plant is going to be affected by the tobacco mosaic virus. A genome to be contains is going to consist of 6.3 to 6.5 kilobase pairs. It has 3' and 5' terminus, where 3' terminus is arranged into a tRNA during the translation, and 5' terminus is going to be methylated guanine, where it's going to be arranged like a mRNA segment. These genes of the tobacco virus is going to synthesize 4 proteins, 2 replicative protein and 1 RNA dependent RNA polymerase for the synthesis, and another is going to be a capsid protein. So which involved in RNA polymerization. Replicative protein is involved in replication process, capsid protein for capsid synthesis. After the genome has been synthesized, we are going to see about each of the protein to be synthesized by the TMV virus. There are replicative protein is going to be synthesized 126 kilodaltons to 183 kilodaltons, or the proteins to be synthesized at the 5' end genome. As we said, a tRNA is going to be made by 3' terminus undergoes folding to 3' terminus undergoes folding to tRNA, whereas the 5' end get methylated. This end of genome is going to synthesize the 126 kilodaltons and 183 kilodaltons of protein, which is involved in replicative action. It involved in RNA capping of mRNA to convert the positive strand of RNA into a mRNA needs RNA capping and unbinding of RNA during replications, both going to be carried out by the replicase protein 126 kilodaltons. Another is 183 kilodalton protein is going to involve in ORF formation, which involve in RNA dependent RNA polymerase action is going to be carried out. Before that, there is a need of ORF formation. ORF means open reading frame. A open reading frame of RNA of a TMV virus is going to be created by this 126 kilodalton protein. After that, the RNA polymerase is going to read and going to add off further the nucleic acids. Therefore, the replication is going to be occurs. Next, a protein of 13 kilodaltons is going to be produced, which is a movement protein. Movement proteins are the protein involved in movement of the nucleic acids in the matrix, so that a TMV virus can move from one plant virus to another plant virus through plasma desmata by the movement proteins. Therefore, this virus can infect more plant cells and produces more progeny of daughter cells, where a core protein is also synthesized, which is a capsid protein. A capsid protein is known as core protein. It's going to be 17.5 kilodaltons. It produces a rigid coat for the virions. It encapsulates the viral RNA. That tobacco virus is going to be a positive sense, which is going to undergo translation and create of its own proteins directly from the RNA, from the original RNA. But why there is a need of RNA dependent RNA polymerase and replicase protein is a question. Because first off, the RNA is going to be a positive sense. By the RNA polymerase, it's going to synthesize its negative sense. Then this negative sense is going to act as a template for the synthesis of more positive sense. When the more positive sense is going to be created inside the plant's virus, inside the plant virus after the infection to be caused by TMV virus. A positive sense is going to create a negative sense is going to create more positive sense. More positive sense is going to express its proteins by using the host machinery, capsid proteins. Then the positive proteins is going to be assembled into its capsid and created a new TMV virus more. Here, a positive sense is only going to act for translation, expression for the protein production. But it was replicated by the RNA polymerase for production of more of positive sense. Thus, for that a replicase protein is used, which is involved in RNA capping. 183 kilodalton protein is involved in binding of RNA polymerase to the RNA positive sense RNA, original positive sense RNA, where movement protein is involved in infecting more plant viruses, where core protein involved in synthesis of capsid.